Now we will take a look at the salts of carboxylic acids. When the proton is removed from a carboxylic acid, the anion is formed, and we call it the carboxylate ion. Carboxylate ion will be weak bases because the acid is relatively strong. And when we name carboxylate ion, we will just change the ic acid ending in the name of carboxylic acid to ate. For example, if we have a sodium salt of acetic acid, we will just name it as sodium acetate. Okay, the ic acid ending is changed to ate. If we have a potassium salt of benzoic acid, we will name it as potassium benzoate. Again, ic acid ending is changed to ate. And the next one, this is the sodium salt of a carboxylic acid with 8 carbon atoms, right? So we name the 8 carbon acid as octanoic acid, and the salt will be octanoate. For this last molecule, something is interesting. It has a long carbon chain, which are relatively hydrophobic, but it also have a relatively polar head, which will be hydrophilic. So what's circled here is what we call a hydrophilic head, and then we also have this hydrophobic tail, or we can also call it lipophilic. For small carboxylates, because it has the hydrophilic head, so they are soluble in water. But as the carbon chain gets longer and longer, that tail becomes less soluble. So something interesting will happen. For the carboxylates with 12 to 18 carbon atoms, if you dissolve them in water, they tend to form what we call micelles, which means these molecules will have their hydrophobic tail pointing to the inside of this spherical structure and the hydrophilic head pointing outside, making hydrogen bonds with water. So the micelle is a colloidal dispersion of spherical aggregates. Typically, it will contain 50 to 100 individual molecules. And micelles will form spontaneously when carboxylate concentration exceeds a certain minimum value, which is called the critical micelle concentration. And again, the polar carboxylate groups dot the surface of the micelle, bind to water or maybe metal ions, and the nonpolar hydrophobic chains are directed toward the interior of the micelle. Induced dipole forces bind them together. They are weak binding forces, but ultimately significant. And the micelles are irregular. It contains voids, channels, and tangled carbon chains. And it can also incorporate some small molecules inside. Well, micelle structures can explain the cleansing action of soap. Soap are actually long carbon chain carboxylates. Soap can remove grease by enclosing it in the hydrophobic-like interior of the micelles, while the grease is washed away by water, not because it dissolves in water, but because the grease dissolves in micelles that are dispersed in water. So soaps are carboxylates, while detergents are the substances that also cleanse by micellar action, and the detergents are actually alkyl sulfates. So instead of having that COO minus in soap, detergents will have SO3 minus. So for example here, sodium lauryl sulfate. Lauryl is dodecyl, which means the 12 carbon chain. So here's the structure with 12 carbon attached to SO3 minus. That's the sodium dodecyl sulfate. It's one type of detergent. It works in the same way as we just described for soap in micelles. But detergents will be especially effective in hard water containing calcium ions and magnesium ions. With these magnesium or calcium ions, soap won't work very well because calcium carboxylate or magnesium carboxylate are insoluble in water. They tend to form soap scum, which will be very disgusting solids formed in bathrooms, etc. But the calcium or magnesium salt of these synthetic detergents, the sulfates, are soluble and retain their micelle-forming ability in water. So for the areas that the water are pretty hard, like in the Midwest area, a soap won't work very well, but instead detergents are good cleaning agents.